Dear colleagues and friends, well, here I am in my home office here recording this talk instead of being at Snowbird at FNANO and enjoying the exciting discussions and posters, beers and skiing with you. Well, thanks anyways for your interest in the work from my lab. For those who don't know me, I am Thorsten Schmidt. I'm an assistant professor in the Department of Physics at Kent State University now. And I'm going to talk about some projects that we did still in Dresden on triangulated wireframe structures from DNA origamis and from scaffold-free single-stranded tags. DNA origami is this method that you probably all know, which allows you to fold a long strand of single-stranded DNA with many short synthetic oligonucleotides into arbitrary two-dimensional structures or three-dimensional structures, as you can see on this slide over here. And these structures were the state-of-the-art and are still the state-of-the-art right now. But towards the end of my postdoc with William Shih, it occurred to me that these are terribly material inefficient ways to construct structures. So in the macroscopic world, engineers would not bundle, for example, rods of steel together to build large towers. And what they do instead is they use they use triangulated wireframe structures, such as this crane over here on the left. And I wanted to build something like this. So the triangles, they are the only geometric primitive that are resistant to shear forces, and you can also find these or similar structures in nature. These are microalgae that have some wireframe structure out of silica. And the first student that walked into my lab and who wanted to have a project was Michael Mattis, and he needed the project, and he was a computer scientist, and I challenged him with this one here: design this, such a crane here, and build this out of DNA. And I also bought him some magnetic toys, such as these ones over here. And playing with these toys, he found out that you can build structures entirely out of tetrahedra. So this one is called a tetrahedral truss. This is a structure that I was not aware of previously, that it existed, and he reinvented that. But if you want to build this out of DNA, it's easier to design this in 2 dimensions and then reconnect these vertices to one another. So we designed this large structure here, and we found a route to route the scaffold through the structure. All of these 6-arm junctions over here, they contain the scaffold here in blue and the stapled strands in these different colors, and they're made out of 12 and 25 base pair domains, and importantly also these 3 nucleotide single-stranded loops over here, which turned out to be very important for the correct folding of these structures, as I'm going to demonstrate later. So as the FNANO is ill-suited for designing these triangulated wireframe structures, we wrote a program, or Michael wrote a program, which allowed the user to just click a route through this mesh over here. And then when you're done, then you can click export to FNANO, and then you can use this design further in FNANO. To cut a long story short, we created 4 different designs. This one is the flat structure, which you can see over here. This one is this tetrahedral truss, which you can see over here. And this one is a dodecahedral truss, which you can see over here. And these structures are cool because they contain much more volume per mass than these other structures. So they're much more material efficient than these structures with hexagonal square lattice, for example. They contain defined cavities, which you can use to fill with nanoparticles or enzymes that you may want to study. And importantly, they can also be folded at physiological salt conditions. because electrostatic repulsions, they are short-range forces, and they don't affect these very hollow structures, not to the same extent as the conventional DNA origami structures. Now I want to talk more about a follow-up project where we wanted to make the construction of these structures even more material efficient by leaving out single-stranded gaps in these structures here that we can then fill with a polymerase and DNTP instead of synthesizing all the bases chemically. This is another representation. So this DNA polymerase is going to extend this one strand all the way to this staple strand over here, and the gray strands, they are the scaffold strands. So we wanted to make this even more material efficient, and we wanted to induce structural transformations. And that is possible because this representation here is a little bit simplified. That is because double-stranded DNA has a persistence length of around 50 nanometer, whereas single-stranded DNA is much more flexible and the persistence length is only around 1 nanometer. So if the single-stranded region here is longer than the persistence length, then there is a force contracting these two ends together, simply because there is much more conformational space here to sample when it's closer together. And also these ends here, they are much more flexible than in double-stranded DNA. To illustrate that, we have recently made some new simulations of this. So here you can see just one unit out of this larger structure here with these 6 triangles over here. This is a simulation that is done in OxDNA in Peter Schulze's lab at Arizona State University. And you can see here these single-stranded regions over here, and you can see double-stranded, single-stranded, and double-stranded regions all over these structures. And when I start the simulation, then you can see that these single-stranded regions here are in fact much more flexible and do contract this entire structure together, and you cannot really see the triangles very well anymore after a couple of nanoseconds of simulation time. In the other case, when you have everything double-stranded, so now these green segments here were freshly synthesized by a polymerase in the experiment. In the simulation, you can obviously not see that. Then you can see that these triangles are much more rigid and are much more intact if everything is double-stranded and they do not contract anymore. So this is the idea. We start with we fold a gap structure, which is a little bit crumpled up, and then extend it with a gap-filling polymerase. For this project, we synthesized 4 different structures. These 2 structures here on the top here, they have gaps of 7 nanometers, and these 2 on the bottom here have gaps of 28 nanometers. These 2 are flat, and these are tubular structures. So, and when you fold these 2 structures with still relatively short gaps, then you can still see that this is probably going to be a flat structure, and this still looks tubular. In the other case, with the very long gaps where 66% of the scaffold is unpaired single-stranded DNA, then everything is contracted to these hair bolts here, which you cannot really distinguish very well from one another anymore. And I should mention that all of these here are the same scale bars, so these are really much more compact than the structures that only have 33% of gaps. But then when you add the gap-filling polymerase and DNTPs within 1 to 3 minutes, all of these gaps here are extended, and you can see the structures are looking as we designed them, and they're indistinguishable from control structures that were folded with the missing staple strands instead of DNTPs and the polymerase. We tested a bunch of different polymerases for these experiments, and to our surprise, a lot of them worked pretty well. T4 is probably the best, but T7 DNA polymerase also worked, and these 3 polymerases that are usually used in PCR reactions and that usually work at much higher temperatures. They also work a little bit slower than usually, but they also work at 37 degrees, and they can also be used for these gap extension reactions here. What did not work was DNA polymerase 1 and these two enzymes here that have a strand displacement capacity. So Klanov and Phi29, they would not stop over here, but they would just push this other staple strand away, and then you just create a double-stranded circle from the M13 scaffold. These reactions, they only cost $1 per reaction to create enough DNA origami to do a couple of gels or to image them in electron microscopy or AFM experiments. So this could be interesting to prototype different versions of DNA origami structures in a very cost-efficient way. We also found that it is very important to inactivate the polymerase after it reached the end here. When you let these reactions run for too long, the exonuclease activity could digest this, or the exonuclease activity could also chew back the strand that it has synthesized when we, for example, load this to the gel. And to prevent this, we added tumor captor ethanol, which denatures the polymerase. And so with inactivation, all the structures, they look pretty good. In some cases, we really saw degraded structures without the inactivation step. And you can also see this very clearly in denaturing page gels when you load these structures and look at the length of the extended staples here. You find that in the paper. So our strong recommendation is to inactivate the reaction after 3 to 10 minutes. So here we looked at the reaction times that were necessary for these extensions, and we found even at lower temperatures, 12 degrees, 3 to 10 minutes are sufficient. And if you leave this for much longer, then the results get worse. So finally, we wanted to play another trick with this gap filling reaction here. So we designed a tubular structure where the inside only contained single-stranded regions, such that the contraction would curve this tube here into a C-shaped tube. You can also see that this also happens in simulations, and not only in simulations, you can also see this very well in experiments over here. Before the extension, all these tubes here were C-shaped, and after the extension, they were all straightened out. These were experiments done by Nayan Agarwal, who is now a postdoc with Ashwin Gopinath at MIT, and Michael Mattis designed all these structures. He's now with Peter Schultz. Now, finally, I want to show you how you can build triangulated wireframe structures without a scaffold, just from single-stranded DNA. And we built the structures that you can see here. This is a paper that appeared last year. And for this structure, we started again with a computer program, which Michael wrote, which allowed you to just click the different triangles in this canvas over here. And then the program would populate the triangles that you clicked with any of two types of single-stranded DNA. So these ones here we called the core staples, and the ones on the outside we called those the protector strands. And the domain lengths were 10 and 11 base pairs respectively. Again, in these experiments, it was very important that we included single-stranded spacers that were unpaired, and so to investigate that further, we just designed this very small structure with 2 rows and 2 columns. That's the 3R2C structure over here, where we only varied the number of thymidines here in the single-stranded spacer region here, so from 0 to 4 thymidines over here. And if you have no spacers at all, everything aggregates and does not migrate into the gel. And only when you start to include 1 or 2 thymidines, you can start to see some product. And only when you have 4 thymidine spacers all over here, then you don't see any aggregation anymore and the formation of the intended structure as the main product. So we wanted to investigate why that is, and we did some simulations again together with Peter Schulz. And in these simulations, we varied the number of thymidine spacers here and ran a lot of these simulations now. And Micha then pulled them together and analyzed them. And if you had no spacers, so 0T spacers, the structure is intrinsically curved and it is a little bit more flat when you increase the spacer length. But the relaxation of the strain by the spacers would not really explain alone why the folding yields were so bad. So instead, we suspected that the folding yields, the bad folding yields, they were caused by these 6-arm junctions over here. And we analyzed those. I want to cut this story short a little bit. The most important part is that adjacent arms of these 6-arm junctions here, they tend to form stacked conformations. So here you can see the degrees between 2 neighbors and the Gibbs-free energy. So we saw an energetic minimum over here for adjacent arms when there is no spacer. And these minima here, they disappear when you increase the spacer length. And we think that these stacking interactions of adjacent arms here, that they are responsible for the aggregation. So we also folded other structures here. So this is a slightly bigger structure than this 4R4C structure. And when you work with this little program that Michael wrote, then you can decide not to use all of these triangles over here and just leave out some of them here and create such a ring type structure here that you can see here. Or you can connect different edges with one another and create, for example, such a tube. And all of these structures, they fold if you have 2 or 4 thymidine spacers with decent yields, and they look very uniform. You can also use these tiles here to create one-dimensional crystals. So you can circularize these here when you just leave out these protector strands. Then A could, for example, connect with 1 over here, but you can also leave out the protector strands on the other sides and allow some polymerization to happen. And we created different designs, and you can see these tubes here that can have many microns in length. But in some cases, they prefer to have these circles. And to be able to prevent these circles from forming, and also to be able to shape the length of the tubes a little bit better, we included some protector strands, so you can see that in the paper. Finally, I want to advertise a postdoc position which I have open in my lab. And in this project here, we want to create a lipid bilayer here with DNA. So this is already published here in 2018. And now we want to use these DNA encapsulated lipid bilayers, as we call them here, to reconstitute integral membrane proteins, which are usually very difficult to study, and it's very difficult to get the atomic structures out of these. And we want to incorporate these in this DNA-based system here for single particle cryo-electron microscopy experiments. And I think this is going to be a very powerful platform. If any one of you is interested in this or knows anyone who may be interested in this, please write me an email. So finally, I want to thank everyone in my lab in Dresden who participated in this research and in other projects that I could not show today. This is a photo of my lab at Kent State University. It's fun to build that up, and I have very talented and very motivated students. If you have any questions on what I just presented, you cannot just ask as it would have been in FNANO. You can write me an email to my email address over here. I want to thank my collaboration partners and want to thank you for your attention, and I really hope that I am going to see many of you again in a year at FNANO 2021 at Snowbird. Thank you very much.